Okay, hello everyone. My name is Lewis Francis. I'm an associate professor in Swansea University Medical School, and I'm going to talk to you today a little bit about gynecological cancers and the work that we do developing novel drug delivery mechanisms, looking at repurposing drugs that have high toxicity or low solubility, and improving bioavailability, improving efficacy using kind of nano and innovative nano approaches. So I'm going to follow on from Maya and from Tom, who have done an excellent job, capturing the impact, capturing the history and development of the field, and really getting across and translating to you guys why we're so excited about the impact nano is already making in the clinic, which Maya captured really well, and some of the cross-disciplinary research methods and development techniques that we're using to try and improve and align with kind of personalized medicine, targeted delivery mechanisms moving forward so that we position nano right at the forefront of the field. So as Maya said, nano is here. Most people now know what nano is. It's been given a massive kind of shop window in terms of the vaccine with the Pfizer vaccine. And the nano that's particularly impactful in ovarian cancer, which is the most prominent gynecological cancer in terms of mortality rate with a very low 5-year survival rate, is pegylated liposomal dox. So I don't have to talk about why we pegylate. We pegylate to improve bioavailability and stability in the bloodstream. And lipodox really has made a huge difference to this very difficult presenting disease. And we can see that if we look at clinical trials. We can see that in a difficult, recurrent, resistant disease and solid tumor with lots of penetration problems, bioavailability issues, getting enough dose of the drug into the solid tumor environment in ovarian cancer is very difficult. Looking at a review of clinical trials with pegylated lipodox, we can see that patients with platinum-free intervals have an extra 20 months of life in their survival curves. And then these recurrent nasty diseases, which are prominent in a disease of interest, if patients take lipodox, they have a huge improvement of 22 months in their survival rate. So a huge impact on nano already. Frames our interest, frames our excitement about what the future may hold. It's now a standard therapy, so it's having impact because of its favorable safety profile and increased efficacy. The future, though, is in terms of combination. So can we integrate non-platinum-based drugs? Can we look at anti-angiogenic, for example? Can we look at different mechanism of action drugs, integrate those and use different therapies alongside lipodox to further improve the efficacy of treatment and clinical management strategies? So we work in reproductive biology and gynaecological to people in Swansea looking at developing innovative diagnostics and therapies in the reproductive space. In order to do that, I'm going to take you through a case study of one of the examples. So we're particularly interested in epigenetic regulatory mechanisms. So epigenetics are reversible, transient mechanisms of gene expression that are linked and intimately linked to the development stages of disease, both in tumorigenesis, so early tumor, and late high-grade serous disease that we see in the example in ovarian cancer. So chemical compounds that directly target these epigenetic processes are around. First generation epigenetic compounds have made it through to clinic for hematological diseases and not so much in solid tumors. So we've been looking at how we can better utilize the positives of targeting epigenetic complexes using first generation drugs by looking at if we package them, we alter PK, PD, alter interaction with cancer cells. So that's the rationale really for the case study I'm going to talk about. And I'm going to give you a whistle-stop tour of some of the preclinical models that we use in the development of nanomedicine. So nanomedicines are subject to the same regulatory approvals. They're subject to the same benchmark statements in terms of characterizing and knowing what your delivery vehicle does, knowing what the physiochemical properties of your delivery vehicle are, what the encapsulation efficiency, the release profile, the dose ranges and dose effects on host cells. So we spend a lot of time developing 2D and 3D in vitro models that reflect our disease of interest, developing the physiochemical characteristics of prominent drug delivery vehicles, and then optimizing those to make sure we get increased penetration into our tumor models, and then monitoring functional biological effects. So I'm going to take you through what we've done with encapsulating SAHA in a Micelle process. So we can now make polymer nanoparticles of different size, different shape, different charge. They're water-soluble fabrication processes. So as Maya said, it's fairly straightforward chemistry. It's a robust, reproducible process. We can characterize the surface using a suite of characterization tools. We can look at the stability and a tailored drug release. And you can see here that we can change the dynamic of the release of the drug over a long period of time in terms of how we can control the dose and exposure of our target cells. So self-assembly, comb shell micelles in an aqueous media, we get really good encapsulation of our drug compounds, and we get prolonged release of those compounds. So what is that doing in our target cells? And we can see here that across multiple cancers using HeLa, cervical cancer, MCF7s for breast cancer, we can see that we can take good images of the cells. We can look at how the cells interact with the vehicle, but we can also look at how the cells react and the fate of the cells post-drug release. And we can see that in the graphs here, the major take-home message, is that we have increased efficacy and increased cytotoxicity compared to the free drug control. And over a prolonged period of time, we see that. You start to see changes at 24, 48 hours, but at 72 hours, we have a remarkable increase in the efficacy of the drug when it's in a nano formulation. Maybe because of increased dose, prolonged release, increased encapsulation, and availability to those cells. How then do we improve that? So we can take the formulation, the micelles, the polymer backgrounds, their chemistry lend themselves to further functionalization. So you can put other moieties on the surface, not just PEG. In this case, we use hyaluronic acid. The ligand for hyaluronic acid is CD44, which most people know is a biomarker for most cancers across a spectrum of solid tumor cancers. They show increased levels of CD44. And quite often that's in type 2, aggressive, resistant, recurrent disease, which are the difficult diseases to treat in that setting. So what we've done is we've taken HA, we've modified our polymer shell, added HA, and seeing if we can target type 2 aggressive disease using CD44 targeting. We again characterize the physiochemical characteristics, again encapsulate SAHA as an exemplar of epigenetic drugs, and we can see that when we encapsulate within our polymer micelles and we target CD44, we get remarkable increase in efficacy, not just in 2D, but in 3D, so we can penetrate 3D models, which show to the surface. They protect their kind of cancer-initiating cancer stem cells, their driver cells within that 3D environment. We can penetrate that, and we can target those, and we show greater efficacy through targeting. So the simple chemistry, the encapsulation, the targeting, gives us new ways to repurpose these drugs such as SAHA that haven't progressed in clinical trials within the solid tumor environment. That brings us on to clinical challenges. Okay, so ovarian cancer is a serious, high-grade serous disease. We know that it has a low 5-year survival rate, and one of the reasons for that is understanding the biology, understanding the boundaries, understanding the challenges in that space. So we've come together with industry partners, with clinical partners, to try and understand the challenges around gynecological disease, comparing oral delivery, IV delivery versus potentially IP delivery, and can we now move on from those fundamental results of lipodox and other flexible polymer systems for nanocarriers, and really tailor those towards these small these metastatic units, these cancer spheroids that disseminate in the peritoneal cavity? Can we understand the acidic flow, all of the fluid that's rushing in from the lymph nodes, and understand how we can really get across those fluid barriers, across those biological barriers, and improve bioavailability to target not only the primary tumor, but these aggressive metastatic spots in the peritoneal cancers that we see. So with a mix of cell matrix, mix of cancer cells, mix of different types of cancer cells, immune milieu. The tumor microenvironment is very complex. So we have a mix in our team of people trying to understand the tumor microenvironment, chemists who are developing nanovectors and trying to tailor those nanovectors towards the tumor microenvironment, and then biologists and pharmacological processes trying to understand new payloads and drug delivery. And we've really got an exciting mix, and it encapsulates what British Society for Nanomedicine is trying to develop, what the community is developing and having an impact already, and pushing the boundaries of what modern drugs and next generation drugs will look like by using the principles and theories of nanoscale delivery mechanisms. So this is an example of a project that we're doing with a local cancer charity in South Wales with 10 of us Cancer Care. So we're taking our base polymer system, we're modifying that polymer system, adding specific principles and specific properties to those polymers so that we can interface with different payloads and interface with different challenges. Okay, so we do it in 2D and 3D. We integrate. We have money to do small animal studies to check if we're doing them in animal, if our incremental increases are working at the clinical interface as well in preclinical models. Okay, so yes, we can deliver. Yes, we can make different polymers that will cross the boundaries and the membranes of different cells selectively, and we can see that with some basic data here across different polymers. Those polymers have been shown to increase clearance through the liver, stop accumulation in the kidney, which is a major problem for drugs and for nanoformulations, and increase bioavailability to our target organs. That's really exciting for us, and moving on into the future, we're now really developing a platform where we can take a spectrum of nanovehicles and a spectrum of drugs, increase the interaction between them, look at chemical material interfaces, have a suite of characterization across different compound libraries, and tailor our offering to the pharmaceutical sciences. So 3D internalization is a standard. We do that by selective uptake and by changes the routes of uptake into cells, and we can affect this complex disease phenotype. So not just cytotoxicity, but also sensitization, reversing epigenetic profiles of disease to resensitize them to platinum-based drugs, for example, and look at combinations. But we can also starting to show now that we can affect motility, we can affect differentiation of cancer cells. So we can really not just look at toxicity, kill, no kill, but sensitization, motility, and confine the disease cells to their local environment and stop this aggressive, difficult to treat disease. Okay, so we have multiple partners, multiple approaches. It's exciting. It's the future. It's multidisciplinary teams. It's working with industry right at the middle of what we're trying to do. Okay. So, with that then, I'd like to draw to a close. I'd like to thank V4, I'd like to thank the British Society for Nanomedicine for asking us to talk about it. I'd like to thank Tom and Maya for setting up my talk really nicely with the background and giving me the opportunity to show some exciting preclinical data and really try and capture how we're trying to develop lipodocs and develop other formulations to complement those approaches into the future and dovetail with personalized medicine. And the slide here just shows very clearly that vaccines, Pfizer, the mRNA-based vaccines really have put nanomedicine on the map again. We're really coming back around, and nanomedicine is the comeback, kind of on the comeback trail, and highlighting its potential impact across a range of diseases with a range of compounds. So thank you, everyone. I hope you're as excited as we are by this community and by the opportunities provided. And we'd be happy to take questions.